Have you ever gone to the grocery store only to come back home and realize you've forgotten a few crucial items? Sure, everyone's forgetful on occasion. However, have you ever gone to the grocery store and suddenly felt confused as to where you were or how to get home? Have you noticed yourself forgetting important dates despite writing them down, or even important people in your life? This type of memory lapse is not typical and may indicate that you are developing dementia.
Dementia is a syndrome that causes progressive deterioration of cognitive functions such as memory, communication, and judgment. Dementia most commonly affects the elderly and can develop due to a variety of diseases and injuries. The most common cause of dementia is Alzheimer's disease, which contributes to 60 to 70% of all dementia cases. Alzheimer's affects over 5 million people in the United States to date. With the rise in life expectancy and a resulting aging population, it is predicted that this number will continue to climb to reach 14 million by 2050. Because the underlying cause of Alzheimer's is still largely unknown, there is no cure for the disease. If the disease is diagnosed early, however, patients and their family members can better manage the symptoms as they progress. There are several known risk factors for developing Alzheimer's, such as family history and possessing the apolipoprotein E4 allele, referred to as ApoE4.
Current neuroscience and epidemiology researchers are searching for other risk factors that may contribute to the development of the disease. Concerningly, there is a rising body of evidence to suggest that an increased incidence of Alzheimer's may be due to what is in the air we breathe.
With the rise of industrialization and mass agricultural practices, humans have been burning fossil fuels and emitting carbon into the atmosphere at an insatiable rate. This has contributed to increased pollutants in urban areas, and these airborne chemicals appear to exacerbate Alzheimer's disease progression.
As levels of airborne pollution rise, we may be giving a breath of death to those predisposed to Alzheimer's disease. There are two primary biological mechanisms that contribute to neurodegeneration and dysfunction in Alzheimer's brains: oxidative stress and the accumulation of amyloid plaques and neurofibrillary tangles.
Mild oxidative stress happens naturally in the body as a byproduct of metabolism through the production of free radicals. Free radicals, also called reactive oxygen species if they contain oxygen, are molecules with a highly unstable, unpaired electron. Reactive oxygen species will steal electrons from other molecules to become more stable, which causes rampant damage to macromolecules like proteins and DNA. Antioxidants like vitamins will neutralize the highly reactive electron to protect the body. However, if antioxidant defenses become overwhelmed by reactive oxygen species, more cellular damage will occur and cause severe oxidative stress. The body will then respond with increased inflammation. If the damage occurs in neural tissue, glial cells will become activated in an attempt to protect and repair the brain. In the brains of Alzheimer's patients, elevated levels of oxidized macromolecules, inflammatory markers, and activated glial cells have all been observed. Amyloid plaques and neurofibrillary tangles are derived from malfunctioning protein processing in the brain. Amyloid beta is a waste byproduct derived from the cleavage of amyloid beta precursor protein, APP. APP is spliced by enzymes, and amyloid beta is produced as a byproduct that is usually disposed of. Amyloid beta is normally cleaved with 40 amino acid residues, which is fairly soluble. However, in aging and Alzheimer's-prone brains, APP cleavage produces an excess of less soluble amyloid beta with 42 residues. Amyloid beta 42 will build up in the brain and form insoluble plaques and disrupt neural communication. In conjunction with amyloid plaques, the microtubule-associated protein tau will become hyperphosphorylated. This will disrupt the typical structure of neuronal membranes, forming neurofibrillary tangles that further disrupt neuronal communication.
In Alzheimer's disease, it is unclear if the formation of plaques and tangles is directly caused by oxidative stress, but the two processes are both predominant features of the disease pathology. It is oxidative stress that in turn links Alzheimer's disease progression and pollution.
Two major pollutants produced by fossil fuel emission are ozone and particulate matter.
Ozone is a highly reactive gas that is formed by both natural and human-influenced processes. Natural ozone in the upper atmosphere is important for protecting the Earth from harmful UV radiation from the sun.
However, ozone produced in the lower atmosphere from chemical reactions between human-produced nitrogen oxides and volatile organic compounds contributes to worsening air quality and smog.
Particulate matter is a complex mixture of airborne particles. Researchers have found that particulate matter with a diameter of 2.5 micrometers, PM2.5, or smaller, is small enough to travel through the airways and reach the bloodstream and neural tissue.
In the nasal passages, PM2.5 can directly contact the olfactory bulb, the only part of the central nervous system that is directly exposed to the external environment. In the lungs, PM2.5 can become embedded in alveolar tissue and then cross into the bloodstream, where it can eventually cross the blood-brain barrier.
Both ozone and PM2.5 can act as reactive oxygen species in the brain to cause oxidative stress. Human population studies have indicated that a greater exposure to these pollutants contributes to cognitive decline and an increased incidence of dementia.
Despite the establishment of air quality standards to protect people from excessive exposure to pollutants, it is estimated that over 100 million people in the United States live in areas exceeding recommended standards for at least one type of air pollutant. This is partly due to climate change causing an increase in global temperatures and affecting wind and pressure patterns that both increase the production and distribution of air pollutants.
It is crucial that we more fully understand the impact of air pollution and climate change on the rising incidence of dementia and Alzheimer's disease.
While human population studies are useful to observe correlations between phenomena, there is no way to carefully control all variables and therefore understand a direct cause and effect relationship.
To better understand the causal effects of ozone and particulate matter on neural degeneration, we will examine two specific animal studies that contribute to the body of research implicating these air pollutants in accelerated development of Alzheimer's disease. In 2010, a team of researchers set out to determine if ozone exposure in rats would lead to oxidative damage, thus affecting both neurogenesis and neurodegeneration. In this study, rats were separated into 5 groups.
The first group was the control group, and rats were exposed to 30 days of clean air. The second through fifth groups were exposed to ozone for varying time periods, 15, 30, 60, and 90 days. Ozone exposure lasted for 4 hours a day with a dose of 0.25 parts per million, simulating levels of ozone observed in highly polluted areas of Mexico City. After the exposure period, rats were euthanized and their brains analyzed for markers of oxidative stress, neurogenesis, cell death, and changes in glial activation. Prior to the collection of the brains, rats also participated in short and long-term memory tasks called the passive avoidance test. In the training task of this test, rats are placed in a box with 2 chambers separated by a door. Animals are first placed in the safe side of the box. After 10 seconds in the safe compartment, the door between the compartments is opened. When the rat eventually crosses into the other compartment, the door closes and a shock is delivered. After the training task, rats are returned to the box for the follow-up retention task after a 10-minute and 24-hour wait period. In this task, researchers measure the amount of time it takes the rat to return to the shock compartment, which is recorded as retention latency.
The results of this study showed a dose-dependent relationship of ozone exposure on signs of neural degeneration.
Analyzed tissue came from the hippocampus, the temporal lobe structure important for integrating memories. In this tissue, markers for oxidative stress, cell death, and microglia activation increased significantly when compared to controls. Neurogenesis, an important mediator of learning and neuroplasticity in the hippocampus, decreased significantly when compared to controls. These results indicate that ozone exposure exacerbated oxidative stress in the rat's hippocampus and caused diffuse damage, which in turn may have caused worsening performance in the memory task. For the passive avoidance task, there was also a dose-dependent relationship of ozone exposure on worsening memory performance. Rats exposed to ozone had significantly shorter retention latencies for crossing into the shock compartment, indicating that they were more forgetful of the shock they had previously received. Rats exposed to 90 days of ozone had the shortest retention time for both the short and long-term tasks.
These results demonstrate the way in which ozone exposure can have a direct impact on cognitive decline. This implicates ozone exposure as a cause for worsening symptoms for people prone to Alzheimer's. To draw a more direct conclusion on ozone and Alzheimer's, the first author from the previous study went on to conduct a subsequent study in 2015.
In this study, researchers followed the same ozone exposure methods and found that amyloid beta 42 levels significantly increased in rats exposed to 60 and 90 days of ozone. This indicates that the oxidative stress caused by ozone exposure contributes to the acceleration of amyloid deposition and thus could contribute to advancing Alzheimer's pathology.
Future research would benefit from using an Alzheimer's rodent model to draw direct conclusions on the impact of ozone on Alzheimer's disease progression. A study conducted in 2017 did just that in analyzing the effect of PM2.5 exposure on cognitive decline. This study was divided into two parts, a human population study and an ApoE rodent model study.
In the human population study, elderly women across the United States between the ages of 65 and 79 participated in annual cognitive screenings for up to 10 years to assess their development of dementia.
The study was limited to women who are at a greater risk of developing Alzheimer's because the study was partnered with the Women's Health Initiative Memory Study.
Analysis of the participants' results included PM2.5 exposure estimates based on their residential location and inclusion of their genotype for ApoE4 alleles. Participants possessing 2 copies of the E4 allele are most at risk for developing Alzheimer's, while participants with 2 copies of the E3 allele are at a lower risk. In the animal study, E-Fad mice carried either the human ApoE3 or 4 alleles, referred to as E3-Fad and E4-Fad respectively. Each type of mouse was randomly assigned to either receive filtered air exposure or PM2.5 air for 15 weeks. As a control for the Alzheimer's disease model, wild-type C57 mice were exposed to PM2.5 air for 10 weeks. The difference in time exposure was due to the ApoE rodent model beginning to experience amyloid deposition after 2 months. After the exposure period, mice were euthanized and their brains analyzed for amyloid plaques, amyloid beta 42, and hippocampal atrophy, amongst other markers for amyloid-caused neuronal damage. The results of the human study demonstrated that residents in high PM2.5 exposed regions resulted in greater dementia incidence. Furthermore, PM2.5 had the greatest accelerative effect of dementia progression for participants who were homozygous for the ApoE4 allele, followed by heterozygous participants.
While PM2.5 exposure affected all participants, it unequally affected those predisposed to develop Alzheimer's, which demonstrates how exposure to pollution could be a significant risk factor for developing the disease.
The results of the animal study drew similar conclusions. PM2.5 exposure increased levels of amyloid plaques and amyloid beta 42 in all E-Fad mice, with a greater increase observed in E4-Fad mice. PM2.5 exposure caused selective atrophy to the hippocampus in all mice, particularly the CA1 region, which parallels observations of human Alzheimer's.
These results indicate that PM2.5 exposure can exacerbate Alzheimer's disease pathology for those most predisposed for the disease, but it can lead less specifically to neural atrophy even for those not prone to develop Alzheimer's. It is possible, therefore, that PM2.5 exposure could contribute to cognitive decline in even those without a predisposition for Alzheimer's.
This may increase the population of people at risk for dementia. Until we better understand the extent to which only Alzheimer's-prone people are vulnerable to PM2.5 versus those of the general population, exposure to pollution may put everyone at great risk of neurodegeneration.
These two studies on ozone and PM2.5 exposure demonstrate the usefulness of studying air pollution and Alzheimer's in animal models. By isolating specific pollutants, researchers can begin to understand the exact cause and effect relationship of pollution exposure and cognitive decline.
There is more work to be done, for example, in the following domains: studying ozone exposure in an Alzheimer's disease rodent model, isolating elements of the APP processing pathway to understand PM2.5's effect, understanding the role of pollution in neurofibrillary tangle formation, and determining to what extent pollution affects cognitive decline in people not predisposed to Alzheimer's. The quantity of human population studies conducted in this field far outnumbers the animal studies conducted.
More animal studies must be conducted to understand the exact mechanism of action of air pollutants on cognitive decline. In considering this, it is important to note that Alzheimer's disease causality is highly multifactorial.
As evidenced by the role of pollution on Alzheimer's progression, several gene-environment interactions impact the development of the disease. While research attempts to understand how specific pollutants affect individual components of the disease pathology, there is still no way to account for all the possible risk factors.
Overly reducing the individual factors that contribute to Alzheimer's development moves away from a holistic approach to treating the disease that is so necessary.
Maintaining an appreciation for the complex interacting environmental conditions that can give rise to disease could lead to a more effective method of disease management for Alzheimer's.
Nonetheless, a greater understanding of the effect of pollutants on Alzheimer's incidence could lead to novel policy and public health initiatives to slow the rise of the disease.
Placing more strict protections against pollutants such as ozone and particulate matter could lead to a global environment that is less likely to exacerbate Alzheimer's pathology.
Additionally, this method of confronting Alzheimer's opens the door to population-wide treatments of disease.
Oftentimes, when a patient goes to the hospital with an illness, treatment methods focus on just helping the individual feel better. However, by initiating countrywide or global policies in an attempt to treat or manage disease, this would set a precedent for treating illness at a population level by managing the disease-inducing environment. By creating an environment that is less likely to trigger disease progression, fewer people will develop diseases such as Alzheimer's, thus reducing the number of individuals in dire need of treatment.
If we can make our air safer to breathe, we can help protect people from Alzheimer's and globally protect the human mind.